You know, creating a new medicine has to be one of the most complex things we do in science. Before you can even dream about a cure, a pill, or a vaccine, you've got to answer one single massive question. When you're looking at a disease in all its complexity, where exactly do you attack? This is the story of that very first make or break step, finding the target.
And the stakes? Oh, they couldn't be higher. This slide right here really shows you the brutal reality of drug development. You might start with, say, 25 different research programs, 25 shots on goal. But over a decade, and after spending more than $1 billion, almost all of them are going to fail. If everything goes just right, you might get one single approved drug at the end of it all. It's an unbelievable process of attrition.
So that brings us right back to the core question, doesn't it? When you're staring down a complex disease, a bacteria, a virus, or even just our own cells going haywire, where in the world do you even begin? How do you find that one specific vulnerability, that molecular Achilles heel, that you can actually hit with a medicine?
Well, to answer that, scientists have to become detectives. And like any good detective, you don't just start searching randomly. Nope. First, you build a profile. You have to know exactly what you're looking for, creating a kind of molecular most wanted list for the key players in a disease.
And that player has a scientific name, of course, a drug target. Put simply, it's just a specific molecule, usually a protein or maybe a gene, that's right at the heart of the disease process. The entire goal is to find a drug that can interact with this target, change what it's doing, and by doing that, treat the disease.
What I love about this slide is how clearly it lays out the golden rule, especially for things like infectious diseases. The perfect target in a pathogen has to be absolutely essential for it to survive. But just as important, it has to be either completely absent or at least look very different from anything in our own bodies. The motto is simple: harm the invader without harming the host.
For decades, scientists have used some really clever, almost old-school detective methods to hunt down these targets. Think of it like classic gumshoe work, but at the molecular level, all about using logic and systematic investigation to narrow down the list of suspects.
So, the first classic technique is a powerful one we can all understand. It's the process of elimination. If you can't immediately spot the culprit, you just start systematically ruling out all the innocent bystanders until only the guilty parties are left.
And in biology, we have a fancy name for this: subtractive genomics. This image shows it perfectly. You start with this huge pool of potential suspects at the top, all the proteins in a pathogen. Then you apply a series of filters, and with each step, you get closer and closer to a small, high-value group of targets. So let's break down how this funnel actually works.
Okay, here's how we go down the funnel. First, you identify all the proteins that are essential for the pathogen's life, all 673 of them in this case. Then, you subtract any that look too much like our own human proteins. Boom, we're down to 351. Next, a big reality check. Can a drug actually stick to these? This drugability filter leaves us with 123. And finally, you make sure the target is present in all the different strains of the pathogen, leaving you with a final high-confidence list of 69 potential targets. Pretty smart, huh?
All right, another classic technique uses a different, but just as powerful metaphor: finding the bottleneck. Every living thing has these complex metabolic assembly lines inside of it. The trick is to find that one station where everything grinds to a halt if it gets blocked. We call this bottleneck a choke point reaction. It's a special kind of reaction in the pathogen's metabolism. It's unique because it might be the only reaction that uses a certain ingredient, or the only one that can produce a specific vital product. The point is, there's no alternate route, no detour. And that's exactly what makes them such perfect targets. If you block a choke point that consumes something unique, that substance builds up to toxic levels. On the flip side, if you block one that produces something unique, you basically starve the pathogen. Either way, it can be game over. This diagram shows it perfectly. A choke point is literally a single point of failure. You don't have to dismantle the whole factory. You just have to block that one critical conveyor belt, and the entire system can collapse. It's an incredibly efficient strategy.
But, these classic methods, as powerful as they are, have their limits. A lot of today's biggest health challenges, like cancer or Alzheimer's, they aren't caused by a single foreign invader. They're way more complex. And for that, we need a modern super sleuth: artificial intelligence.
We've really moved beyond that simple one gene, one disease idea. We now understand that so many illnesses are the result of these subtle, complex disruptions across huge networks of genes and proteins inside our own bodies.
Finding a single target in that web is, well, it's like trying to find a needle in a haystack that's made of other needles. And this is where AI, specifically something called machine learning on graphs, just shines. As a 2020 paper put it, target identification is probably going to be the greatest area of opportunity for this kind of AI. It is a tool that is perfectly matched to the complexity of the problem.
Because AI can see the whole picture. It can look at a network like the one you see here, this mind-bogglingly complex map of biological interactions, and it can actually learn to spot the patterns. It can identify the most influential nodes, the hidden masterminds that are secretly orchestrating the disease from deep inside the network.
And the clues it uses are incredibly diverse. We're talking about AI algorithms sifting through data on how proteins interact, known links between genes and diseases, these huge databases called knowledge graphs, and even predictions about what molecules can do. It takes all of that information, synthesizes it, and pinpoints the most promising targets.
So whether you're using classic gumshoe methods or advanced AI, the investigation leads to a primary suspect, a validated drug target. But what happens next? What's the verdict?
Well, the verdict is that the real work is just beginning. Identifying that target is a monumental achievement, for sure, but it is not the end of the story. It is, however, the absolutely essential first chapter that everything else is built on.
After a target is identified, that's when the long road to a medicine really begins. Scientists screen thousands of compounds to find a hit. Then they optimize that hit into a lead. Then they do years of preclinical lab testing. And only then, if everything still looks good, do they start the long, expensive, and incredibly difficult process of clinical trials in people.
And this brings us right back to why that first step is just so critical. The entire billion-dollar, decade-long journey rests on the foundation of that initial target. If you get the target wrong, if you aim at the wrong molecule, the entire structure will eventually collapse, often after hundreds of millions of dollars have already been spent.
So it all starts with finding the right place to begin. And that leaves us with a pretty fascinating question, right? As our detective tools get exponentially smarter, which of the diseases that we consider untreatable today will finally have their secrets unlocked tomorrow?